Hello, everybody. Over the next few minutes, we will discuss bone marrow biology that is relevant to the imaging of bone metastases. So we will discuss the cellular content and distribution of the normal marrow and the regulation of the bone structure in order to develop an understanding of the biological mechanisms responsible for the spread of tumors to the bone marrow space and the formation of bone metastases, and we will briefly look at the biological mechanisms responsible for osteoblastic and osteolytic metastatic deposits. Now we know that bone marrow makes about 4% of the body mass and is incredibly active hematopoietically. The normal bone marrow is established by about 25 years, with red marrow being in the axial skeleton. But we also know that there are subsequent changes that are related to age that occur in older age groups, with loss of the normal trabecular bone and loss of synthetic function that occurs later on in life, with the deposition of reticulin and collagen into the bone marrow space. The bone marrow is a complex and poorly understood organ, with stromal cells and the matrix providing the structural scaffold for the hematopoietic cells. There are hidden spaces, these osteoblastic and vascular niches along the endosteal surfaces and around blood vessels for hematopoietic stem cells, but these are also where tumor stem cells reside, and the mesenchymal stem cells are more evenly distributed within the bone marrow space, and these are shown here in green. And in this diagram we can see these mesenchymal stem cells within the bone marrow space, which then become osteoblasts, fibroblasts, and fat cells, whereas hematopoietic stem cells, I said, were around blood vessels along the endosteal surface, and these hematopoietic stem cells give rise to other cells such as megakaryocytes, red cells, white cells, etc. There is a cellular compartmentalization of the bone marrow, with erythrocytes, macrophages and their precursors being found around blood vessels. Granulocytes and their precursors tend to be found at the bone interface. Now we know that bone marrow fat is important, making up about 70% of the bone marrow space. And bone marrow fat plays an important role in energy storage, endocrine function, and bone metabolism, but bone marrow fat is involved in the metastasis process also. There are two types of fat cells that we need to consider. These smaller fat cells, which are found in metaphyses of the long bones and in the central skeleton, which are active metabolically and so are involved in hematopoiesis and skeletal modeling. But there are also yellow bone marrow fat cells, which are much larger size, and they tend to be found in the shafts of the bone marrow, where there is relatively little hematopoiesis going on. And you can see how metastatic disease tends to go mostly to the axial skeleton, with relatively little disease in the shafts of the bone marrow. And when you do see it in the shaft of the bone marrow, you can see that it's up against the endosteal surface. So bone marrow fat cells serve as a lipid source that can be utilized by the cancer cells and help to proliferate and regulate the cancer cells. Adipocytes around metastatic colonies also promote osteoclastic action, enabling bone metastases to enlarge by bone delamination. There is a complex signaling going on between the derivatives of mesenchymal stem cells and hematopoietic stem cells. Specifically, I'm referring to osteoblasts and osteoclasts, which we need to consider next. Now, this appears to be a rather complex picture, but there are 3 or 4 things that you need to take away from this diagram. First of all, hematopoietic stem cells eventually lead to the formation of osteoclasts, and osteoclasts cause bone resorption. Point number 2, mesenchymal stem cells lead to the formation of a number of cells including chondrocytes, adipocytes, and osteoblasts. But it's important to remember that adipocytes and osteoblasts have a common progenitor. The maturation of osteoblasts uses something called the Wnt pathway, and that eventually leads to osteoblasts. And remember, osteoblasts form bone. Osteoblasts mature into osteocytes and then eventually mature into the lining cells on the endosteal surface. And the last point to note is that osteoblasts and osteoclasts signal each other through a mechanism using OPG and rank. So this is shown more simply in this diagram. Mesenchymal stem cells, common progenitor, pre-osteoblasts, all can mature into adipocytes. The osteoblasts mature using the Wnt pathway. Osteoblasts eventually lead to lining cells, or can become osteocytes. The osteoblasts are intimately involved in the formation and the maintenance of the osteoblastic niche. And in the osteoblastic niche, you have these hematopoietic stem cells. We talked about all of these aspects previously. Hematopoietic stem cells lead to the formation of osteoclasts. Remember, osteoclasts are involved in bone resorption. There is signaling between osteoclasts and osteoblasts using this mechanism that we discussed briefly, rank and rank ligand with OPG. So if you want to simplify this even more, we can say that mesenchymal stem cells using signaling molecules lead to osteoblastic recruitment and activity, and that is what is responsible for bone formation. Hematopoietic stem cells through signaling molecules result in osteoclastic recruitment and activity, and this is responsible for bone resorption. And remember, osteoclasts and osteoblasts interact with each other, and increased OPG leads to bone formation, increased rank ligand leads to bone resorption, and it is this mechanism that is hijacked by tumors, leading to osteolytic and osteosclerotic metastases. But before we go into this mechanism, we need to discuss how tumors go from their primary site into a metastatic bone marrow site. Cancer cells have to undergo an epithelial mesenchymal transition, and this enables survival within the bloodstream, but also promotes the transendothelial migration from the blood vessels into the bone marrow space. If you see tumor cells within the bone marrow space, these are called disseminated tumor cells, or DTCs. These DTCs hone towards the endosteal surface using calcium and pH gradients, and these DTCs then compete for the endosteal niche, displacing the hematopoietic stem cells that are sitting there. Once they are sitting in the niche, they take on stem cell-like properties. So these dormant cancer stem cells have a limited cell cycling and self-renewal and therefore are chemo-resistant. Eventually, these cancer stem cells come out of the osteoblastic niche and then form the metastatic deposits by co-opting the function of the osteoclasts and the osteoblasts. And this is shown pictorially here. So here you can see a dormant tumor stem cell in an osteoblastic niche where it has already displaced the hematopoietic stem cell. Once it activates and begins to multiply within the bone marrow space, it initially signals osteoclasts using the rank ligand and OPG mechanism that we discussed. This causes bone resorption and the release of calcium and growth factors, which promotes tumor cell growth within the matrix adjacent to a bone trabeculae. The proliferating tumors require its own stroma on which to proliferate, and it does that by signaling osteoblasts through the Wnt pathway. The Wnt pathway results in the formation of osteoid, and this osteoid is woven bone, which then becomes mineralized. So you've got these two pathways going on at the same time. You've got osteoclastic recruitment and activity, usually around the margins of metastatic colonies, resulting in delamination of bone and growth factor release that promotes growth. And on the other side, you've got osteoblastic recruitment and activity, which forms these osteoblastic lesions through the mineralization of woven bone. So in fact, both processes are occurring at the same time. Not only are they occurring at the same time, but they are not well coordinated. And this leads to different appearances at different times. So this is the same patient a year apart. You can see that on the right-hand side, he's got mostly osteosclerotic metastatic deposits with small areas of osteolysis. But a year later, you can see he's got mostly osteolytic metastatic disease. The other thing you'll see is that I've represented a small colony of metastatic disease as an area of osteoid in the middle, which is calcified due to the deposition of hydroxyapatite crystals, with tumor cells that are surrounding this area of woven bone and the delamination of the bone marrow occurring around it due to the participation of osteoclasts and fat cells. And in fact, you can see this on a CT scan. So if you look at the CT scan, you can see there's an area of dense nidus mineralization in the middle of a metastasis, around which you can see a small halo. And this halo, it contains highly cellular material, as you can see on this high p-value image, which has a low ADC. So let's conclude here. There are three important learning points. First of all, there's a complex and balanced signaling occurring between mesenchymal and hematopoietic stem cells and their derivatives within the marrow space. Cancer cells within the bone marrow co-opt these normal cellular mechanisms to create microenvironments enabling tumor cell proliferation and metastasis expansion. And it is the balance between osteoblastic and osteolytic mechanisms that determines the final nature of the metastases that we observe on bone scans and CT scans. I hope you found this lecture useful and insightful. Leave me some comments. Thank you very much.